Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous videos, we have we have completed the anatomy and the morphology of alimentary canal. We discussed about different parts of alimentary canal, stomach, esophagus, large and small intestine. Today we are going to start with the last part in the morphology and the anatomical section of alimentary canal, and then we will start with the physiology of digestion, means the chemical process of digestion. So we have to start with today pancreas, which is commonly known as sweet bread. Pancreas, it develops from endoderm. The origin of pancreas, which is a gland, it develops from endoderm. It is a soft, lobulated and elongated organ when having a lobules like this, these lobules like this, and it is elongated. Okay, it is made of numerous acini. Acini, they are the working units of pancreas, right? Acini is a group of secretory cells surrounding a cavity. Okay, so acini, they are the working units of pancreas. They are a group of secretory cells surrounding a cavity. Each SNI is lined by pyramidal shaped cells. They are lined by pyramidal shaped cells. These acinar cells secrete the enzyme of pancreatic juices. Okay. So the SNI, they are exocrine in function, mean they basically are related to the secretion of enzymes, pancreatic juices or pancreatic enzymes, right? Now, each SNI opens into a pancreatic duct. Each SNI, it opens into a pancreatic duct. Okay, the duct is basically a fine ductule. Okay, so each SNI opens into pancreatic ductule. These pancreatic ductules, they combine with millions of pancreatic ductules. They combine to form the major pancreatic duct, which is known as what, duct of Wirsung. So SNI, they basically merge to form pancreatic ductule, and these pancreatic ductule, they further combine to form pancreatic duct, okay, which is known as what, duct of Wirsung. Understood? The main pancreatic duct join with the bile duct. The main pancreatic duct joins with the bile duct. Okay, it joins with the bile duct to form hepatopancreatic ampulla. So pancreatic duct plus the bile duct, pancreatic duct plus the bile duct forms hepatopancreatic ampulla. It forms hepatopancreatic ampulla. Okay. Now, this is a mixed gland. It is a mixed gland when it works as both exocrine and endocrine function. It is a mixed gland. It is both exocrine and endocrine in function. Exocrine and endocrine in function. Right. Understood? Now, the exocrines, they are having duct, and endocrines, they don't have duct. I mean, they don't have duct. Okay, their secretion directly comes into the blood. Okay, so the endocrine glands, their secretion come directly into the blood, and the exocrine, they pour their secretion into the duct, and the ducts, they deliver the secretion into a particular target organ, right, into a particular site. Understood? Morphologically says the pancreas, it is having the head, body and tail. Okay, I mean the three regions, like this is the head part, this is the body part, and this is the tail part. Understood? Now, hepatopancreatic ampulla, which opens into the duodenum. Hepatopancreatic ampulla, it opens into the duodenum. The accessory pancreatic duct, known as what, duct of Santorini. Duct of Santorini opens into the duodenum. Opens into the duodenum with separate opening located above the opening of major pancreatic duct. Okay. So hepatic pancreatic ampulla, it opens into the duodenum. The accessory pancreatic ducts, they are known as what? Duct of Santorini. They opens into duodenum with separate opening, okay, located above the opening of major pancreatic duct, okay. And the duct of Wirsung is basically the main pancreatic duct, which is known as what? Duct of Wirsung. Some group of endocrine cells are also found in between groups of acini. These group of cells known as what? Islets of Langerhans. These islets of Langerhans secrete insulin and glucagon hormone. Some group of endocrine cells, they are also found, they are basically found in between the groups of SNI cells. So these intra-acinar cells, they are known as what? Isles of Langerhans, and these isles of Langerhans, they secrete insulin, glucagon hormone. So this gland is exocrine as well as endocrine in function. It's 99% part is exocrine, while 1% part is endocrine. So it is the heterocrine, mean the exocrine and the endocrine function, they have differences, mean exocrine is more dominant function wise on the endocrine part. So they are heterocrine, they are mixed glands, they are heterocrine. In rabbit, the bile duct and the pancreatic duct both separately open to the duodenum. Okay, in humans, the bile duct, it merges with the pancreatic duct to form the duct of Wirsung or the major pancreatic duct, which opens into the duodenum along with the openings of accessory pancreatic ducts known as what? Duct of Santorini. While in rabbit, the bile duct and the pancreatic duct, they open separately, open into the duodenum. The bile duct opens into the proximal limb of duodenum. It opens into the proximal limb of the duodenum and is controlled by sphincter choledochus. Sphincter choledochus. So bile duct, it opens proximally into the limb of the duodenum. It opens proximal, or opens into the proximal limb of the duodenum, means the anterior most part of the duodenum, and is controlled by sphincter of choledochus. Pancreatic duct opens into the distal limb of the duodenum called sphincter pancreaticus. Okay, so there are two regions at the front, mean at the forward part, at the front part, at the anterior most part, the bile duct opens, which is controlled by sphincter choledochus, and the posterior part is for the pancreatic duct in rabbit, for it opens to the distal limb of the duodenum, sphincter pancreaticus. In humans, both bile duct and pancreatic duct combine to form the common duct known as what? Hepatopancreatic duct. The terminal end of the common duct is swollen and is called ampulla of Vater or hepatopancreatic ampulla. Ampulla of Vater opens to the middle part of the duodenum and is controlled by sphincter of Oddi. Bile duct is controlled by sphincter of Boyden. Okay, so the sphincter present in the bile duct is Boyden. B for bile duct, B for Boyden. Sphincter of Boyden, बताओ बताओ. Sphincter of Boyden, bile duct. Okay, sphincter of Oddi, which is controlling the opening of ampulla of Vater into the duodenum. Okay, so I'll recapitulate all the points for you. In case of humans, the bile duct and the pancreatic duct, they don't have separate openings into the duodenum. Okay, I mean, we do have only a single opening, which is the opening for the major pancreatic duct, that is known as what? The hepatopancreatic duct. The terminal part of this hepatopancreatic duct is swollen, and this swollen up part is known as what? Ampulla of Vater. I mean, this is a physiologic swelling. It is not inflammatory, neither it is infectious. It is a normal physiologic swelling, okay, which is known as what? The hepatopancreatic ampulla, right? Now, this ampulla, hepatopancreatic ampulla, is known as what? Ampulla of Vater, which opens to the middle part of the duodenum, and it is guarded by, it is regulated by the sphincter of Oddi, right? While the bile duct for is guarded by the sphincter of Boyden. Understood? Is that clear? Yeah. Now, so before starting with the physiology of digestion, I would like to, comment some more points about this gland. Okay. Now, the exocrine part is basically of the pancreatic acini, and endocrine part is basically because of the islets of Langerhans. Now, there is very important question. Now, the secretion of bicarbonate ions in pancreas is secreted by bicarbonate ions is basically they are enzymatic in function. Their main function is to buffer. So the bicarbonate ions secreted by HCO3 minus, secreted by acinar cells. They are secreted by acinar cells. Okay. Now, the endocrinal region or the islets of Langerhans, they are basically having three regions, or the three types of cells. The alpha cells, which are arranged on the peripheral margin, then comes the beta cells, okay, and then comes the delta cells. So alpha, beta, and delta cells. The endocrine region in the endocrinal part. Okay, so the endocrinal part is having alpha, beta, and delta cells. Okay. Alpha cells, they have different function to secrete glucagon. Beta cells have different function to secrete insulin, and delta cells to secrete somatostatin. In delta cells, they inhibits the insulin and glucagon secretion. Okay. So alpha cells, beta cells, and delta cells. The functions I will be, considering later on in this coming video. Okay. There is some important cells known as what? Pancreatic peptide cells, PP cells. PP cells. Pancreatic peptide. Pancreatic peptide cells. These pancreatic peptide cells, they act on the pancreatic SNA and inhibits the secretion of pancreatic juices. Okay. So the pancreatic peptides, they are the additional cells which are present, the PP cells, the pancreatic peptide cells, along with alpha, beta, and delta cells. Alpha cells, they are concerned with the secretion of glucagon hormone, beta cells for insulin, and delta cells, it further regulates the secretion of insulin and glucagon by the secretion of somatostatin. The somatostatin, it works on the feedback mechanism of controlling the or regulating the secretions of the glucagon and insulin. Okay. Now coming to next, physiology of digestion. Physiology of digestion, I will be considering all the process of chemical digestion, functions of enzymes, bile juices, everything will be detailed discussed under this section. The physiology of digestion says what? The first is the digestion of food in oral cavity. Food enters through mouth. Okay, when enters through mouth. Okay, when the part where ingestion happens, the food is tasted in oral cavity and mixed with saliva. It mixed with saliva. The tongue mixes the food with saliva. Tongues help in the mixing of food with saliva. This food with saliva is called, it is known as what? Bolus. The saliva, when the pH is 6.8 to 7, contains water 99.5% and electrolytes Na plus, K plus, Cl minus, HCO3 minus, and thiocyanate, SCN minus. Okay. So food enters through mouth, food is tasted in oral cavity mixed with saliva. Tongue mixes the food with the saliva. It helps. When tongues help to push the food so that the food comes in between the oral masticatory process. Okay, because the upper jaw and the lower jaw, they occlude. Okay, they match the cusp of the teeth. They matches the grooves of the lower teeth. Okay, or the cusp of the lower teeth, it matches the grooves of the upper teeth, and they occlude well, so that mastication can happen, okay, to its maximum. So the food of saliva is known, the food with saliva is known as what? Bolus. And the salivary pH is 6.8 to 7, mean mild acidic. It contains water 99.5%, electrolytes Na plus, K plus, HCO3 minus. HCO3 minus, it act as a buffer. It act as a buffering agent. It acts as a buffering agent. Cl minus, thiocyanate. Chemical digestion. In this type of digestion, saliva act with food particles. Saliva contains 99.5% water and 0.5% salts. It contains 99.5% water along with 0.5% of salts. These salts are organic as well as inorganic. These salts are organic as well as inorganic. The main salts are mucin. The main salts are mucin, okay, thiocyanate. These salts like sodium thiocyanate, okay, sodium chloride, sodium bicarbonate. The main salts are NaCl, sodium thiocyanate, okay, then potassium chloride, then sodium bicarbonate. All these salts are there. Okay, these salts get ionized, and especially the bicarbonate ions, it helps in the buffering agent, mean it helps to maintain the pH of saliva. Helps to maintain the pH of saliva. Maintains the pH of the saliva. Okay, now mucin, which is mucus secretory cells, then lysozyme, which is antibacterial. Lysozyme, which is antibacterial. Mucin, which helps to, which helps to secrete mucus. Then ptyalin is an enzyme, salivary amylase. Okay, so the first process, of the chemical digestion or the physiology of digestion is ingestion, which is intaking of food. Then mastication happens with the help of teeth, tongues, saliva. Teeth chew the food, and then the physical breakdown of the mechanical churning, mechanical mastication happens. Then deglutition means, well, swallowing of food. Deglutition, the food enters into the, it enters into the pharynx part. Okay, it enters into the esophageal part. So deglutition, the nerve centers which control the deglutition process, okay, they are located in the medulla or medullary regions of the brain. Okay, so deglutition center is located in the medulla. Okay, then food bolus is pushed by the tongue. Okay, then involuntary muscles in the pharynx are stimulated. The involuntary muscles of the pharynx, they get stimulated after the initializing of food bolus into the food esophagus. Okay, so the tongue pushes the food bolus into the esophagus or the pharynx. Okay, involuntary muscles in the pharynx are stimulated. Now the larynx, it moves up, okay, during the process of, ingestion, mastication, deglutition. After deglutition, the moment food enters into the esophagus, the larynx moves up. The epiglottis, it swings and covers it covers the glottis, okay, which is the opening of the windpipe, and the opening of food pipe or the esophagus is gullet. The esophagus swings and it covers the glottis. Okay, soft palate, and uvula, it closes the internal nostrils. Soft palate and uvula, it controls the internal nostrils. Upper esophageal sphincter relaxes and bolus moves into it. Okay, so this is the whole journey I'll like to tell you, okay, before going to all these. Okay, just a minute. Yeah, so what basically the first thing is ingestion. The first thing is ingestion, right? It is ingestion, which is intake of food. Then mastication, in which mechanical breakdown or physical breakdown happens of food takes place. Then deglutition, swallowing. The center is located in medulla. Okay. Then the food bolus is pushed by the tongue. Okay. During the process of deglutition, the food bolus is pushed by the tongue. Okay. Now it enters into the pharynx. It enters into the pharynx. After entering to the pharynx, the larynx goes up Okay, the larynx goes up. Epiglottis covers the glottis, which is the opening of windpipe, and the food enters the esophagus. The upper esophageal sphincter relaxes. Okay, so the upper esophageal sphincter and the food bolus comes into it. Okay, soft palate and uvula, it close the internal nostrils. Soft palate and closes the internal nostrils, right? Yeah. Now, so the ingestion, mastication, deglutition, the food bolus is pushed by the tongue. It enters the pharynx. From pharynx, it pushes up the larynx to go up. Okay. The epiglottis, it covers the glottis. The upper esophageal sphincter, it relaxes. The soft palate and the uvula, it closes the internal nostrils. Now, coming back, mucin. Mucin is a glycoprotein. Mucin is a glycoprotein, right? It lubricates the food particles. It helps in swallowing of food. Mucin is a glycoprotein. It lubricates the food particles and it helps in swallowing of food. Lysozyme, it is an enzyme which kills the harmful bacteria. Due to this reason, saliva is antiseptic in function. Thiocyanate, it is a special salt which kills the harmful bacteria. So it is called bactericidal salt. Thiocyanate is bactericidal salt. It kills the harmful bacteria. Lysozyme, it kills the harmful bacteria. So it is a type of antiseptic lotion. Okay. Then Tylen, starch maltose plus limit dextrin. So Tylen is basically a salivary amylase. I mean it converts starch into maltose. Okay, plus limit dextrin. It converts starch into maltose. Starch into maltose mean it breaks starch into maltose plus in limited amount dextrin is formed. Tylen is found in human saliva because human food is mainly made up of starch. Tylen digests only ripe and cooked starch. It does not digest the starch. Okay. In case of humans, the food is mainly having the fibers or the starch which is cooked. Mean already initially it is broken down. Okay, the polymer, the bonds of the polymer of the starch, it is broken down because of heat. The Thailand, it digests only the ripened and the cooked starch. It does not digest actual the raw starch. Thailand is absent in saliva of rabbit and carnivores animals because food of rabbit is mainly made up of cellulose, so there is no function of Thailand over there. So Thailand is absent in the rabbit, mean core herbivore animals, and especially the carnivore animals, they don't have ptyalin because there is no need to digest starch, right? Deglutition peristalsis. Bolus is pushed inwards through the pharynx into the esophagus. This process is called swallowing. It is coordinated by the activity of the tongue, soft palate, pharynx and esophagus. The tongue blocks the mouth. Soft palate closes off the nostrils and the larynx so that epiglottis closes off the trachea in the windpipe. The food moves downwards into the esophagus. A traveling wave of constriction called peristalsis pushes the bolus of the food downwards into the esophagus. Peristalsis is produced by the involuntary contraction of circular muscles, which is preceded by simultaneous contraction of longitudinal muscles and relaxation of circular muscles lining the gut. When a peristaltic wave reaches the end of the esophagus, digestion or digestive enzymes are absent in esophagus. The sphincter opens, allowing the passage of bolus of the food into the stomach. Gastroesophageal sphincter of the esophagus and stomach normally remains closed and does not allow the contents of the stomach to move back. When reverse peristalsis is protected by the gastroesophageal sphincter or the lower esophageal sphincter. Okay, the lower esophageal sphincter is the gastroesophageal sphincter only. There is no difference. Okay, so please remember both of these names, right? So the bolus of the food is pushed inward through the pharynx into the esophagus, and this process is called swallowing. It is coordinated by the activity of the tongue, soft palate, pharynx, and esophagus. The tongue blocks the mouth, soft palate closes off the nose and larynx, so that epiglottis closes off the trachea. Food moves downward into the esophagus. The traveling wave constriction called peristalsis pushes the bolus of the food downward. Peristalsis is produced by involuntary contraction of circular muscles, which is preceded by a simultaneous contraction of longitudinal muscles or of circular muscle lining the gut. When a peristaltic wave reaches the end of the esophagus, digestion and digestive enzymes are absent in esophagus. The sphincter opens, allowing the passage of bolus of food into the stomach. Gastroesophageal sphincter of the esophagus and stomach normally remains closed and does not allow contents of the stomach to move back when reverse peristalsis. Yeah, you can see the relaxed muscles, circular muscles, they contract. Constriction, it makes a passage. Okay, it makes a way for the pushing down of bolus of the food. Okay, longitudinal muscles, they contract, and because of the shortening of the passageway ahead of the bolus, longitudinal muscles, they contract. They again create a pressure. Okay, the circular muscles contract. Gastroesophageal sphincter is closed. Okay, relaxed muscles, the circular muscles contract, constricting the passageway and pushing bolus food. Okay, then the longitudinal muscles, they contract, shortening the passageway ahead of bolus food so that food can be pushed again. Then the wave of the contraction, it can generate it. Okay, so this is the way peristalsis happens. Peristalsis and segmental movements, these two types of movements you will come across, and there is a difference between peristalsis and segmentation. Peristalsis, the adjacent segment of the alimentary canal organs alternatively contract and relax. Food is moved distally along the tract. Very important. The distal movement of food is seen. Okay, primary, propulsive, some mixing may occur. Okay, the primary movement is propulsive, and then some mixing may occur. Non-adjacent segments of the alimentary canal organs contract and relax. Okay, mean the non-adjacent segments of the alimentary canal or organs, they contract, but the adjacent don't. Food is pushed or moved forward and then backward. First comes forward and then backward. Primarily mixes with the food and breaks it down mechanically. Some propulsion may occur. Peristalsis, the involuntary contraction and relaxation of muscles of the intestine, creating wave-like movements, pushing the contents of the canal forward. The back and forth smooth muscle contractions, mainly in the small intestine for the smooth muscle contractions. Main occurs in esophagus, mainly occurs in the small and large intestine. Okay, occurs by the contraction of circular muscles, occurs due to the rhythmic contraction of the longitudinal muscles, a one-way motion in the caudal direction responsible for the movement in both the directions, allowing the greater mixing, results in high speed propagation of food, results in slow propagation of food. So this is the difference between peristalsis and segmentation. Peristalsis mainly involves the contraction of circular muscles, and the segmentation involves the contraction of longitudinal muscles, right? So this is the difference. Yeah, so that will be all for today. Till then, bye bye, take care everyone, stay safe, stay healthy, bye.